Cells don't divide all the time. The timing and regulation of the cell cycle is currently one of the most exciting fields in cell biology because of the relationship of cell cycling to cancer. This is video 10.4 about some of the basic mechanisms that make sure that a cell divides only when it's supposed to. We'll cover these 4 unit goals. If you want a closer look, pause the video now. In one of our previous videos, we talked about the relative timing of each phase of the cell cycle, where the cell cycle spends most of its time in interphase, but this video is more about what controls the frequency of cell cycling. In our bodies, different cell types are needed in different ways, so the different cell types each have different frequencies of dividing. For example, early in development, an embryo invests a lot of energy into growing and growing and growing, so embryonic cells are among the fastest dividing, where they divide about once every 20 minutes. That's incredible. In contrast, liver cells only undergo mitosis about once every year or two. Some cells, like muscle cells and neurons in your central nervous system, don't divide at all. They're arrested in a phase of the cell cycle called G0. When it's time for a cell to divide, there are definite stop and go signals to make sure everything goes right. You can roughly classify these signals into external signals, signals from surrounding cells that typically start the cell cycle, and internal signals, signals that are inside the dividing cell that act as checkpoints along the way. The external signals we'll talk about are growth factors, anchorage dependence, and contact inhibition. The specialized proteins called Cyclones, CDKs, and P53 are the internal signals we'll focus on. Anchorage dependence means that a cell's ability to divide is dependent on the cell being anchored or attached to a surface. This can be seen in this diagram when cells are added to a petri dish. The cells won't actually start dividing until the cells are attached to the dish's surface. If a cell becomes detached or unanchored, the cell will stop dividing. Another related idea is contact inhibition. Let's check out this diagram in more detail. When cells are put into a petri dish, the cells divide and fill up the dish completely. When the cells are touching or contacting other cells on all sides, the cells stop dividing. In other words, cell division is inhibited by contact with other cells. If some cells are removed by scraping, this means that some cells on the border of that hole are no longer in contact with other cells on all sides. So the cells at the fringes here will start dividing again until the plate is full. And once this plate is full, contact inhibition kicks in and the cells stop dividing. Another external regulator for cell cycling is growth factors. Growth factors are ligands, signaling molecules that signal for a cell to start dividing. Just like other signaling molecules, growth factors are made by one cell, the signaling cell, and bind to a receptor on another cell, the target cell. Hopefully you're looking at this diagram while you're listening, and some wheels are starting to turn here. You see a dimerized, phosphorylated receptor, which means that we're looking at receptor tyrosine kinase signaling. If you follow the relay molecules in response, you'll see that this signaling pathway activates CDK, that special protein that's an internal signal of cell cycling, and leads to accelerated entry into the cell cycle. Here's another example of growth factor signaling. The growth factor ligand is in the growth factor receptor, and in this case, there's a kinase cascade that takes the signal into the nucleus. Once in the nucleus, the cyclin genes are turned on, leading to cell division. You'll notice that these last 2 slides had cyclins and CDKs, those specialized internal signals for cell cycling. This is the point where the signal goes from external, using growth factors, to internal, relying on CDKs and cyclins. Growth factors are the go signals to start cell cycling. Once the cell cycle starts, however, the pace of the cell cycle is set by CDK and cyclins. Moreover, there are 3 checkpoints of the cell cycle along the way, shown here by these gates, 1, 2, and 3. Well, let's take a closer look at CDKs and cyclins and see how they work. CDKs and cyclins are both proteins. CDK stands for cyclin-dependent kinase, and that name tells us a lot. First, we can tell that CDKs are enzymes, based on the ACE name. Next, we can tell that CDKs speed up phosphorylation reactions because that's what kinases do. The things that CDKs phosphorylate are other proteins that have to do with cell cycle related jobs, like making the mitotic spindle or condensing the DNA. The last thing we can tell about CDKs is that they depend on cyclins. CDKs will only phosphorylate things if a cyclin is attached to the CDK. So if CDKs are like quarterbacks in football, then cyclin is kind of like the football. Just like a quarterback can't do their job without the football, a CDK can't do its job unless the CDK is holding a cyclin. As you can tell by the IN in the name cyclin, cyclins are proteins. The levels of cyclins fluctuate throughout the cell cycle, and cyclins have the job to bind to CDKs and activate CDKs in 2 ways. First, cyclins change the shape of the active site of CDKs. And second, cyclins help take CDKs to the exact spot in the cell where the CDKs are needed. In other words, cyclins are allosteric activators of CDKs. Cyclins bind to a site away from the active site, but change the shape of the active site, allowing the substrate to fit in. There are different types of cyclins for different stages of the cell cycle. The main cyclins are cyclin D, E, A, and B. You can see on this graph that the levels of each change throughout the cell cycle. Cyclin D levels increase just to get the cell cycle started in the G1 phase of interphase. Then during G1, levels of cyclin E increase as this protein is transcribed and translated. The peak of cyclin E expression signals the transition from G1 to S phase of interphase. And during S phase, cyclin E is degraded, but cyclin A gets turned up. When cyclin A is high, there's a transition from the S phase into G2 of interphase. During G2, cyclin A is degraded and cyclin B builds up. The peak of cyclin B expression signals the transition from interphase into mitosis. What's crazy about cyclins and CDKs is that they're so highly conserved. Every organism that does mitosis has CDKs and cyclins, which means that these proteins are really important and haven't been dropped over evolutionary history. What this also means is that you can take a cyclin or CDK gene from one species, like yeast, and put that gene into another species, like humans, and the gene will still work. Crazy, huh? Where CDKs and cyclins do all of their work is at the 3 cell cycle checkpoints. These checkpoints are the G1S checkpoint, the G2M checkpoint, and the M checkpoint, also known as the spindle checkpoint. At each checkpoint, imagine the cell cycling being momentarily stopped to double check that critical things have happened. Then the cell can move on to the next phase if things are okay, or the cell can stop if things aren't okay. Stopping is called cell cycle arrest. When a cell is in cell cycle arrest, the cell can either fix the problem and then move on, or the cell can die by a process called apoptosis, which is when the cell intentionally dies or commits suicide. Here's a more detailed view of the checkpoints and what's being checked. At the G1S checkpoint, growth factor signals are needed to get the cell cycle going. So we want to check for those growth factor signals. During G1, the cell has been growing, so the cell size is checked to make sure the cell is big enough. Also, The status of nutrients is checked to make sure that nutrient conditions are favorable to complete the whole process of cell division, because why get started if you can't finish? Next is the G2M checkpoint. DNA synthesis just happened during S phase, so it's this checkpoint at the G2M checkpoint where DNA replication, we want to make sure that that whole process was completed and that any mutations have been corrected before moving on. The final checkpoint is the M checkpoint, also called the spindle checkpoint. The main question here is, are all of the chromosomes properly aligned at the metaphase plate? The first checkpoint is the G1S checkpoint. Here, cyclin E is paired with its CDK, and those are in charge of making sure the cell size is big enough and the nutrition status is favorable. If everything is good, the cell will progress into the S phase. If there's something wrong, cells enter a phase called G0, which is cell cycle arrest. Now, G0 isn't necessarily a bad thing. G0 just means that a cell isn't dividing right now. G0 can mean that a cell is doing its normal job and working and will divide later. G0 can mean that the cell cycling is paused while the cell is being fixed. Or G0 can mean that the cell won't divide because it just doesn't need to. The second checkpoint is the G2M checkpoint. Accumulation of cyclin A and its associated CDK are the molecular aspects of the G2M checkpoint, and this checkpoint makes sure that DNA replication has been completed and that any mutations have been found and fixed. If these requirements are met, the cell can continue. If not, it's off to G0 to get fixed or get killed. Cyclin A and its associated CDK are sometimes called MPF, for mitosis promoting factor. Some of the jobs of MPF just really prepare the cell for the dance of mitosis that's coming up. And these jobs include phosphorylating proteins involved in chromatin condensation. Let's pack that DNA so it's ready to move. Disassembling the nuclear envelope. Let's get the nuclear envelope out of the way for the DNA that's going to move. And forming the mitotic spindle for all of the jobs that the spindle does. With a little bit of understanding under our belts, I think we should revisit how CDKs and cyclins work. Check out this graph that shows the level of CDKs and cyclin A over time, the two components of MPF. The green line shows that the level of CDK remains constant over several cell cycles. That's the CDK corresponding with the green line. The blue line that shows the levels of cyclin shows that cyclin A increases, then decreases, then increases, and decreases. In other words, cyclins get their names because their levels cycle. So CDK remains stable over time, and cyclin A increases and decreases with each cell cycle. Remember, the increases happen by increasing transcription and translation of cyclin A, and the decreases happen by degradation of the cyclin A protein. Now, also remember that CDKs can only do their job when there are cyclins around to act as allosteric activators of CDKs. In other words, CDKs are always there, just not always on. The last checkpoint is the M checkpoint, or the spindle checkpoint, where cyclin B and its CDK are the stars of the show. Once the chromosomes are checked to be lined up at the metaphase plate, the CDK is involved in activating things like kinetochore motors and a complex called APC, or anaphase promoting complex. That's shown right here. Check that out. That's a huge complex of proteins. And some of those get phosphorylated by the cyclin B associated CDK. Anyway, among the jobs of APC are degrading the proteins that keep the sister chromatids together, and that allows the chromatids to walk their separate ways towards the centrioles during anaphase. The last aspect of cell cycle regulation I want to talk about is a protein called P53. P53 is kind of like the police of DNA damage. Don't think of P53 as being part of any specific phase of the cell cycle, but as being on the lookout at all times for DNA damage. If P53 detects any DNA damage, P53 will first act to stop cell cycling. Next, P53 will initiate the mechanisms for DNA repair. P53 doesn't repair the DNA itself, but kind of lets other proteins know that DNA repair is needed. Finally, if the DNA isn't fixed, P53 will signal the cell to undergo apoptosis, or programmed cell death, also known as cell suicide. Think of P53 as the cell cycle enforcer. Here's a diagram showing how that works. The orange circle is the site of DNA damage. In this diagram, it's saying that the damage is from heat or radiation or chemical damage. P53 detects this damage and stops any cell division. Next, P53 triggers the activation of DNA repair enzymes. If the DNA is repaired, then the cell can move on. But if the DNA isn't fixed, P53 triggers the cell to self-destruct by apoptosis. Then the cell turns into a bunch of fragments that are then cleaned up by macrophages. So let's recap what we covered in this video. Cells can use external and internal signals to help regulate the frequency and the pace of cell cycling. External signals that we talked about in this video include contact inhibition, anchorage dependence, and growth factor signaling. Once the growth factor signals are received, the signals go from external to internal. CDKs and cyclones are the main internal factors that we talked about that help pace cell signaling at the 3 checkpoints. CDKs are kinases that are allosterically activated by cyclone binding, so those two work together. The 3 major checkpoints that we talked about are the G1S Checkpoint, the G2M Checkpoint, and the spindle checkpoints. Finally, P53 detects damaged DNA and triggers the mechanisms to cause that DNA damage to be repaired or triggers the cell to die.